Yeah. So as I told, we are going to continue the last part here, which is about the excretory system. So let's start the excretory system. And in this chapter, and on actually all of your bio topics, I have told you this earlier that everything we will be covering first in plants, and then we will be covering that in animals. So let's start. I'm going to share my screen. So you see my screen. It's a presentation. I'm putting it on the slideshow. I make sure you just see the screen only. So let me just cross check it again. Okay. Okay. So basically, guys, we are going to cover these topics. Excretion in plant, and then we will be covering excretion in animals, and then we will be talking about related to the excretory system in human being, what issues we face. Yes, we are going to discuss a little about the kidney failure. So the first thing that we have to talk about is excretion. Guys, even before I talk about excretion in plant, let me tell you one thing. Excretion is the way of removing waste product. This is like, if I talk about the general description, this is like all the metabolic waste products that mainly include the nitrogenous waste, they are thrown outside the body. We do have the digestive system, that's the digestive waste. Metabolic waste is in the cells, all the reactions are happening, all the cycles are happening. So whatever be the waste, metabolic carbon dioxide and ammonia and urea, these all nitrogenous waste, amino acids, ketone bodies, everything is just removed. That's I'm talking about in animals, but in plant also there's a metabolic waste. We have to be discussing about it. So if I talk about in general, it include mainly the respiratory waste, metabolic waste I'm talking about. And then it include the nitrogenous waste, as I mentioned. And then, yeah, it also consists of the bile waste, bile pigment in human being I talk about. Otherwise, these waste, they're toxic. They're going to create harm for our body. So that has to be removed. So let's talk about excretion in plant first. Excretion means removing the waste. I am repeating this thing. Now let's talk about the plant, how excretion happen. First, stomata, all the removal. As an excretory waste, you may say even CO2 is released. And it is not so that into the plant that waste is getting removed from the stomata. It just released like transpiration I was talking about. Here also carbon dioxide, any kind of gaseous waste that is getting released. So it is also in the leaves, as in stomata, in the stem, as in lenticels, I repeat, as in roots, as in, there are different type of roots. Actually, there's some modified variety of the roots which are little outside, they are producing the waste. So these kind of waste, and through general surface also, because carbon dioxide can also come out. You must have heard about gums and resins in the plant. That is also a type of waste which is stored inside the plant, and that is excreted out later after. That is also stored in the parenchymatous tissues, if you remember tissue. Anyways, gums and resins. Then sometime there are little crystals also that are deposited into the plant. It appears to be like made up of calcium oxalate. This is also into the plant, which is type of waste. Every year, plant used to shed up the leaves. That's also way of just removing the unused part of the plant. Anyways, now if I talk about in individual cells, in an individual cell of the plant, there are vacuoles where this excreta waste is stored. Guys, I tell you one more thing. You must have seen these part of the plant. See, if you're talking about the waste, first you have to talk about all the stomata, lenticels, bark, and everything. Now you must have seen this thing. This is actually not the water dews. It looks beautiful, but that is also a way of removing the waste from the plant. This appears to be like a dew of the plant. This phenomena is known as guttation. And actually onto the leaves, there are little pores present at this place where it is happening. That pore is known as hydrothores. Through the hydrothores, water which is mixed, waste which is mixed in water, this is getting excreted out. So that's the diagram. Hydrothode is there. So it is also protected by the guard cells, but inside there's a little air chamber where all the fluid is getting to be out. And these are all the gums and resins in the plant. So this is how excretion happen in the plant. They also excrete. It's just that they have a different type of waste, but their waste are useful for us. This resins and gums, they are used in varnishing and paints industry. So I suppose that's clear to you. Now we have to talk about excretion in animals. Talking about excretion in animals. First of all, excretion in animal happen by different ways. It is not so that we have excretory system including kidney and all. No, we excrete in different ways. Suppose I'm saying there are sweat gland present on my skin. They are also removing the waste. I am excreting out carbon dioxide by breathing. That's also waste. On the skin also, sweat and other salt and urea, uric acid, nitrogen of waste is released. Is that clear? Other than that, there is a specific excretory system also. You know, excretion in animals happen by different ways. Talking about us, no, talking about the picture that you see on the screen, I'll explain that first. So on the basis of what kind of waste any organism is producing. Proteins are made up of amino acid, nucleic acid are made up of nitrogenous bases. It's a part of it. Further, they modified to form the amino, they are having amino group, and what amino group they are having on the basis of that, they all are nitrogenous. This is also nitrogen compound, this is also nitrogen compound. It's just that depending upon their structure, what is being secreted out in different organisms, these organisms are categorized into three categories. So first is the one which is releasing ammonia as a waste product. These organisms are known as ammonodelic. Ammonia is not, it's very pungent. It's not very, a good chemical. It's very harmful. That's why most of the aquatic animals, they include, they excrete ammonia, which is dissolved in the water. So most aquatic animals, including many fishes. Talking about the urea. So mammals, us, amphibians, and shark and some bony fishes, they excrete urea. They are known as ureotelic. Ammonia, urea, uric, they all are made up of nitrogen. They all are having a amino group, but they are nitrogenous ways. Their structure is different. And the way it is also present is different. Like in water, it's a gaseous form. Ammonia is little dissolved in water. Urea appears to be like, the urine. Uric acid appears to be like the condensed structure, a little paste kind of, because in birds, you must have seen that. So yeah, uric acid. These are known as ureotelic. Now, I suppose that's clear to you. Let me mention some invertebrates also. We do have the excretory system, which are made up of the kidney, right? So we do have the excretory system. We have kidneys and ureter and urethra, whatsoever. I'm going to mention that. But in invertebrates, there are many animals who have different ways of surviving. Like I suppose I talk about amoeba. It has contractile vacuole, which helps in excretion. Is that clear? Now, if I talk about some insects, they have a tubular-like structure, which is known as Malpighian tubules. Yeah, that's Malpighian tubules. Actually, Malpighian tubule appear to be like a tube kind of structure, and it is present nearby, connected to their digestive system. But that helps in excretion and excreting the waste product. Now, let me talk about Platyhelminthes category. Simply, we call it as the flatworm. They have a little cellular structure. We call it as the flame cell. It appears to be like, this is also known as protonephridia. Protonephridia. It appears to be like a little flame, like, it has cilia. The way it works, it beats like a flame, and that's why it is known as flame cell. Other name, protonephridia, present in the flatworms. Now, I suppose that is clear to you in another organism. Now we have to finally talk about human. Let's move forward. Why do I have a blank space here? Okay, I'm going to write something. So basically, talking about the excretory structure present in our body. First is the kidney. Then we have the ureter. Then we have the urinary bladder. Damn, on the blank space, you can just write in large fonts. Urinary bladder, urethra. That's it. These all are working in a proper way. Into the kidney, filtration of blood is happening. Our excretor system is present on the lower abdominal region. Getting my point? Second, blood from the heart, like from the aorta, blood is being flowing into all of the part of the body. Then there is aorta. We call it as abdominal aorta, which is going into the abdomen, the largest aorta. This is further making branches, giving blood to the kidney. And kidney may what happen? Filtration happen. So the urine is actually technically a filtrate. It must be, na? Okay. Now let me think something else. One more point I would like to add, not here, but on this screen. As you see, this is the basic excretory structure. You people must have been seeing since your 6th grade, but it is time to understand it in a clear way. So basically, this is aorta, red one. Obviously, red indicates oxygenated one, blue indicate the blue, which is deoxygenated one. One thing that you have to understand, artery. It consists of oxygenated blood plus the waste product, nitrogenous waste. Vein. Excuse me. It consists of deoxygenated blood plus no waste. You understand my point? My point is aorta is the one that is coming toward the kidney for filtration. Purification of blood in human body happen at two places. One into the lungs for oxygenation. Heart is just pumping the blood from the heart is going in the lungs. So one in the lungs where the exchange of gases happen, oxygen and CO2. Second in the kidney where the wastes are removed. So the artery is the one which is having oxygenated blood. It comes into the aorta, through the aorta, towards the kidney. After filtration, it is moved back. So artery is the one which will further be going inside and it will be forming the blood vessels, arteries, arterioles, capillaries. So ultimately into the nephron, capillaries will be the one where filtration happen because capillary is the only place where exchange happen, right? So into the kidney, that story happen. After that, filtrate through ureter come out. I'm going to explain you about the kidney and the structural part of it, but before that, understand through the ureter it's coming out and the urine will be collected into the urinary bladder, which is a muscular organ. In a day, we excrete through nearby 1.5 to 2 liter of urine, and it's pale yellow. I have told you that why it is pale yellow. It has a pigment which is known as urochrome, which is a leftover pigment of the hemoglobin. That's why it's pale yellow. And yeah, now let me tell you about the bladder. Talking about the bladder, it doesn't have so much of the capacity. It's like nearby 300 ml of something. When it starts to fill up, it's going to send signals. Actually, the muscles of the urinary bladder is known as detrusor muscles. They are elastic muscles. They can increase the size. So when it starts appearing to be half a full, yeah, it starts sending signals to the brain. It's time to urinate, and after a while you can just hold, but when it's getting full and full, you cannot hold them. So it's half when you just get enough. So anyways, this is a cone-shaped structure, urinary bladder, which is elastic. It can increase the surface area. And you must have heard that urination happen more in pregnant women because nearby the ureter, urinary bladder is present and the size of the urinary bladder is increasing and it's putting a pressure on the urinary bladder. So that's the reason that, they cannot uphold a good amount of urine. But second point is I'm talking about the urinary bladder here, the detrusor muscles. And the lower part is known as trigone. It's the bladder neck. This is the internal urethral sphincter. This is external. And that's the urethra. One thing. If a question come in a paper, what is the difference between male and female reproductive system? Sorry, I'm mentioning third chapter here. But one thing you will be talking about the urethra. Because into the male, this is only penis through which the reproductive fluid is coming out. Also, this urine is also coming out. So, and in women, the reproductive system and the urethral opening is different, which is common in male. That's what you have to remember. And the length of urethra is also, of course, long in male and it's less in female. So it's about the urinary bladder. Let's move forward. Yeah, it's a kidney. Bean-shaped organ. You remember? So it appears to be like a purplish brown structure. Its weight is nearby 150 gram. It is having nearby 12 centimeter in length. Yeah, and about 6 centimeter thick, or widthness, you may say. 6 centimeter widthness. Kidney. It is the one that is filtering. What is inside the kidney which is filtering it up? Blood capillaries are in contact with what? Yeah, I know that you know the answer. It's nephron. It's fine. I'm going to come to that point. So this is a kidney. It is having different segments in it. These segments appear to be like a triangular shaped structure like this. And this triangular structure, this is known as renal. Yeah, they have not written the word renal. That's renal pyramid. So the triangular structure is known as renal pyramid. Guys, I don't know whether you remember or not, but in bio, there are two general terms. One is known as cortex and one is known as medulla. Cortex is usually labeled for something which is external part, and medulla is something which is internal part, like the central one. So the cortex part, did they mention? Yeah, this is renal cortex, and then did they mention renal medulla? This is renal medulla, the central part. This is the central part, and this is actually the cortex part. It appears to be like most of it. This is the cortex part, I will say. Now, guys. Okay, so renal pyramid, they are meeting up at a central point. This one that appears to be like a wheatish. Is that a wheatish color? No, it's a creamy, pinky, creamy, something like that. So this is known as renal pelvis. So point is that the central part through which where all this renal pyramid are getting connected, this is known as renal pelvis, which is ultimately going toward the ureter. And this concave structure here, this is known as renal hilum. Hilum is just the concave structure present. Concave structure. Concave is not a word. Okay. So now let me tell you how nephron is present. Into this, first of all, the blood vessels are coming. They are making all the capillaries. Through the artery, they are coming, and through the vein, they are moving back. Clear? Now, ultimately, in this, nephrons are present. There are millions and millions of nephrons present inside the kidney. This nephrons, it appears to be like a tubular structure like this. A hell lot of tubules like capillary, in the same way, a little tubular structure. Not little, a lot. And half of them is present in cortex. Not half of them, I will say half part is present in cortex and half is in the medulla. Like some part is in the cortex and some part is in the medulla, like that. So there are millions of nephrons. So ultimately, these nephrons will be having a collecting duct, because into the nephron, urine is being filtered, blood is being filtered. So they are having a collecting duct which will be meeting up into the renal pelvis. And from here, the urine is collected and then we excrete out. It's a gradual process. Fair enough. Now, I think it is a time to understand the structure of nephron. So take a look. Here it is. I'm going to draw it. I'm bad in drawing, but that's fine. I'm going to make sure that you understand. So point is that the two most important part into the nephron is known as Malpighian corpuscle. And the second one is renal tubule. Tubule is tubule. Malpighian corpuscle further used to have two parts, Bowman's capsule and glomeruli. It's fine, you're hearing technical terms. I'm going to explain, don't worry. Renal tubule is further of three types, PCT, LOH, and DCT. It's fine, I'm going to tell you the full form once I show you the picture. So these are the parts. Point is that one thing that you have to remember, that into the nephron, blood is being filtered. So blood is having lot of, into the nephron, blood is being filtered. So nephron is having a lot of blood vessels nearby it in the form of capillaries. So if It is a nephron. It'll be having all blood vessels around it, covering it up. So it'll be like a mess. Let's a little fold and defold it up. Firstly, it appears to be like a cup-like structure. We call it as the Bowman's capsule. Okay. So then there is a tubular structure. We call it as the renal tubule. And this will be connected to the collecting duct. Now let me draw it. Dear, I'm trying to make it best, but the diameter is less. In this area, the diameter is comparatively a lot less than the one that I'm drawing. So I'm going to make it broader from this. Yeah. This is all the tubular part. So what is the Malpighian corpuscle? This is Bowman's capsule. Let me just change the pen color. So basically, I told you blood vessels are present everywhere. First of all, blood vessel is coming here and it is going to coming back. Make it little less diameter. The one which is coming is known as efferent blood vessels, efferent vessel, and the one which is going is efferent. Efferent is RE, efferent is JARE. Remember that way. Efferent vessel. So inside what happened, the blood arteriole is coming here, efferent one. It is making a network because obviously it will be making a capillary. So it will make a huge network of capillary here. So this capillary network is the one which is known as glomerulus. It is actually nothing but the capillary network. Bowman's capsule is the part of the nephron and the glomeruli is actually the blood vessel itself. Together they are known as Malpighian corpuscle. So glomeruli is not part of the nephron, it is a blood vessel. Together they make Malpighian corpuscle. Now, this part, this is the tubular part. This whole structure is a tubule. This is known as PCT. We call it as proximal convoluted. Convoluted as in there are convulsions, foldings. Convoluted tubule. And this part with the less diameter, this is known as loop of Henle. LOH. And the one where suddenly diameter increases again, this is known as DCT. Distal convoluted tubule. Proximal is pass wala, distal is dur wala. Proximal is proximal, distal is distal. Okay. So here from the blood vessel, blood is coming and this blood is going to be filtered into the glomeruli and then filtrate will be entering up into the tubule. Into the tubule, urine will keep on moving because blood has been filtered and it will keep on exchanging the molecules, waste product throughout. And this is the collecting duct. The collecting duct, it is having thousands of nephron connected to it here and there. And then ultimately, this is the one which will be going toward the renal pelvis, if you remember the diagram. And that will be going to the ureter and urinary bladder and urethra and out. Okay. Now, one thing that there are many things striking up my mind right now. One thing that you have to remember, guys, I told you that microvilli is present in the digestive system, which is increases surface area. FYI, microvilli is also present in proximal convoluted tubule. They also increase surface area. They also help in maximum absorption. So this is the point that you have to understand. This is the structure of the nephron. I'm going to explain you how nephron work, but let me tell you one additional thing. I'm going to make it with green. Okay. Actually, I'm going to show the blood vessels. So let's select the green color. At least select. Anjali Ahuja. Yeah, done. Sorry. So as I told you that this nephron, it's covered by the blood vessels. I'm going to make this diagram mess, but you make it double layered, kind of, tubular kind of. Blood vessels are present everywhere around it. everywhere. It's just covering it up. See, I'm just cutting it up. This blood vessels, this is covering the tubule everywhere. The time when you make it, please make it tubular one. I have made it like a single line. This blood vessels nearby the loop of Henle. The blood vessel which is present nearby the loop of Henle is known as vasa recta. Otherwise, blood vessels are known as peritubular capillaries. Peritubular. So many students actually get confused at this point. Let me tell you, all the blood vessels present nearby the nephron is known as peritubular capillary. And the one which is present nearby the loop of Henle, it is known as vasa recta. It is also peritubular capillary, but the name is vasa recta here. So I suppose the structure of nephron is clear to you. Now let me tell you, half of the nephron is present in the cortex part and half will be present into the medulla part. There are different type of nephron on the basis of which part in that's not a point of discussion, but for now you just remember. So you can actually write it in the diagram that this is medulla and upper one is cortex, and it is having again Malpighian tubule, Bowman's capsule and glomeruli. Glomeruli is nothing but it is a group of blood capillaries. Okay, capillaries with a single P, not the double one. So PCT, LOH and DCT. Here urine will be moving, not urine, not blood, we call it as the filtrate, because blood is being filtered through the capillary, it's coming into the Bowman's capsule. One thing I would like to add. Okay, I would like to add here. If you see the structure now, how simple and beautiful diagram, but I made it good, huh? Okay, so this one, the one which is coming is efferent and the one which is going is efferent. Why did not they mention it? Doesn't matter where you want to make it, above or lower. But inside, this is the glomeruli, glomerular capsule. This is the Bowman's capsule. Acha, they have mixed it together, glomerular capsule. This is the glomeruli, this is the Bowman's capsule. So they have mixed it up. Now guys, into the glomeruli, there are many cells present which are helping the blood to filter. Those cells are known as podocytes. I repeat myself, there are a group of cells present inside the tubule. This is known as podocytes. Here blood will be filtered. Now, anyways, this one is proximal convoluted tubule. Did they mention it in the diagram? Yeah. This one is proximal convoluted tubule. And then there is a loop of Henle, which comparatively less diameter. You see, loop of Henle. And then there is the distal convoluted tubule, the rest of it with a bigger diameter. This is collecting duct. This part is cortex, and this one is medulla. This is cortex medulla junction, and all the blood vessels and all the nephron part is covered up by the blood vessels. Peritubular capillary network is covering it all around, and the one which is present nearby the loop of Henle, we also call it as the vasa recta. But even if you don't remember the vasa recta term, you simply write at the capillary complex. Why blood vessel is present nearby it? Because they keep on exchanging the material. That's how filtration happens. Useful material from the tubule go into the blood vessel, and the waste material from the blood vessel go into the tubule back. So one story happened here. Second story happened here where actual filtration happened. This is actually the mechanism of urine formation, the one that I'm explaining right now. If you want to learn that in great detail, okay, here it is, mechanism of urine formation. Give me a second, guys. Hello. Hold on for a second. Okay. So am I clearly audible to you? Oh, I don't know what just happened. Somebody just joined my link. Okay. So let me just share my screen with you again. What just happened? I'm like surprised what happened. Okay. So now I was explaining you mechanism of urine formation here. Into the mechanism of urine formation, 3 steps happen. First is glomerular filtration, second is tubular reabsorption, and third is tubular secretion. So first is glomerular filtration. Here into the glomeruli, filtration happens through the blood vessel. Urine is being filtered and all the filtrate come into the tubule structure. And this process is, I call it as the ultrafiltration. You can also call it ultrafiltration. Now, after that, after ultrafiltration happen here, this is going to further move. Second is tubular reabsorption. Tubular reabsorption means if you see the second arrow, red one, it is moving toward the peritubular capillary. So what happened? All the waste product, all the useful product is moving back into the useful product is moving back into the blood vessels. Are you getting my point? That include the sugars, amino acid, whatsoever, minerals, potassium, calcium ions, or not calcium, yeah, potassium and sodium ion, they're moving back. Now, the third one is tubular secretion, where the waste product from the blood come back. One thing that you have to remember, ultrafiltration happen in Bowman's capsule. Tubular secretion, the second step, it used to happen mainly in PCT and loop of Henle, and mainly tubular secretion happen in DCT, where the waste material from the blood vessel is secreting out back. So that is what the mechanism of urine formation. If that question come in your Olympiad, I suppose you'll be able to write, and even if in your board exam. With this, we are done with this excretion mechanism. Now we have to move toward the last topic of the excretion system, which is kidney failure and hemodialysis. I have seen a case in my life. One of my cousin was there. He was probably 17 years, I guess. Yeah, he actually, survived for exactly 9 months. And after that, he got died because of the kidney failure. It did not match with any of the person nearby. So kidney failure is like when kidney is not able to filter. What if we filter it out? That person was also on this machine. Obviously, it has certain limit. The machine cannot hold you throughout the life. But my point is that whatever the time it is, we can just go. I have another, known person who survived for 3 years, and she was of 65 age. So that's what I want to say, that this is actually an actual machine. We call it as the hemodialyzer, where we are taking up the blood out and we are filtering it up. You getting my point? How we filter it out? Let's understand. Hemodialysis means separating the particles. We take the blood out from the renal artery, we filter it up, move it back to the renal vein. You're getting my point? This happens in a machine which is known as dialyzing machine. It consists of a filter in it, which is a cellulose fiber. So, sorry. It's a nitrocellulose membrane. Nitrocellulose membrane. So into this nitrocellulose membrane will be there. It will be filtering up the blood. So first of all, blood from the patient, it enter into the dialyzer here, and internally filtration happen. After filtration, move back into the body. There are certain things that we have to take care. Externally, their temperature has to be maintained as like a body temperature. We have to add anticoagulants like heparin. Anticoagulant with the C. For example, heparin. What I'm writing, Heparin. Heparin. So here, blood is, could you just give me a second? Guys, give me a second. There's some person joining my session. I have to tell them to stop and do not enter my session. Okay, let me share my screen again. Okay, drop the message. So basically what happened, we have to maintain the temperature. Second, we have to add the anticoagulant. Through the nitrocellulose membrane, blood is being filtered out, it's moving back. The solution that we used in this machine, that is known as dialyzing solution, where there's like a movement of the dialyzing solution from down to up, and the used solution is thrown out here, dialyzing drain. And this is the fresh we keep on adding. So it's a filter, blood is being filtered, and yeah, dialyzing remove waste product from the blood by filter. And movement of the blood I've shown you, purified blood. Yeah, so purified blood is moved back. When we are moving the back, we have to take care of this. If a question come in your paper, what are the criteria that we have to follow during the time when we do hemodialyzing? One, temperature. Second, anticoagulant. Third is that there should not be any bubble. There are actually bubble trapper that we use as a machine. They trap the bubble because a person can die because of attack or something if there's even a small little bubble entering into the body. So that thing is well maintained, and blood continuously go through the vein, and that's it. This is hemodialysis. Now, if you see this picture, that's the same process I have explained. With this, we are done with the excretory system of the life process, and neglect the thing that happened today's session. It was my colleague, he by mistake entered my session, but that's fine. You're anyhow learning the class. Okay, how is oxygen and carbon dioxide transported in human being? I have explained you this. Transport of gases, I don't have to explain anything on this. So you can pause the video and read it. I'm moving. What are the components of transport system in human being? Blood and heart, blood vessels, lymphatic vessel also. About the lymphatic vessel, guys, if a question come in your paper about the function, you write it in the same way. Lymphatic system also help in the transport. It also help in thermoregulation, osmoregulation, transporting minerals, molecules, and everything. That's common, like the same way you write. I mean, about the blood, but in the lymphatic system, you can mention about the lymph, how it is protecting. Okay, so finally, we are done on the excretory system. And I suppose it is clear to you. I'll see you up in the next chapter probably. Till then, you study well. And then if you find any doubt, you have to post it on the forum, the discussion board. And if you have any doubt more, ask your teacher in the class. Bye-bye.